A snake can cross desert sand faster than most animals with four legs. No arms, no legs, no external ears, no ability to regulate its own body temperature. And yet, snakes became one of the most successful predator groups on Earth. They exist on every continent except Antarctica, in deserts hot enough to melt skin from stone, in rainforests where sunlight never reaches the ground, in rivers, caves, oceans, and trees. Some can swallow prey larger than their own heads. Some can detect body heat in complete darkness. Some carry enough venom in a single bite to stop a mammal's nervous system almost instantly. And for more than 100 million years, they have survived nearly every catastrophe this planet has produced. But snakes were not always like this. There was a moment deep in Earth's history when the ancestors of every snake alive today still had legs, not symbolic remnants, real legs, functional limbs attached to bodies. Some of those early snakes lived while dinosaurs still dominated the planet. Some kept traces of their limbs for so long that modern pythons and boas still carry tiny skeletal remnants hidden inside their bodies today. And what makes this strange is that evolution almost never gives something up without a cost. Legs were one of the greatest advantages vertebrates ever evolved. They allowed animals to run, climb, hunt, escape, and spread across the planet. Snakes lost them anyway, not suddenly, not cleanly, but through a transformation so unusual that even now, snake embryos still begin developing legs before the process abruptly shuts itself down. Somewhere in deep time, an animal crossed a threshold that changed the future of predators forever. If this is your kind of thing, hit like and subscribe. It costs nothing and it keeps this channel going. And drop a comment below. Are you watching this winding down for the night or watching this during your active day? Now, let's go back to the moment snakes still walked on four legs. Long before modern forests filled with birdsong, before mammals inherited the quiet hours after sunset, a lineage of scaled reptiles was already spreading across the planet in silence. They belonged to a vast reptilian group called squamates, animals covered in overlapping scales that glimmered softly under ancient sunlight. Today, this group includes iguanas, geckos, monitors, skinks, and every snake alive on Earth. The division between snakes and lizards is far less distant than it first appears. In evolutionary terms, snakes emerged from deep within the lizard family itself, carrying with them an ancestry that still lingers beneath their altered bodies. The earliest squamates appeared roughly 240 million years ago during the Triassic period, while the first dinosaurs were only beginning their slow rise into dominance. Earth then was warmer, harsher, and crowded with evolutionary experiments. Continents were joined into enormous land masses, and reptiles occupied ecological spaces that would later belong to creatures not yet imagined. Somewhere within this sprawling reptilian world, one branch of lizards began drifting toward a radically different form of movement and survival. Exactly where that branch separated remains one of evolutionary biology's longest running debates. Fossils alone provide only fragments. Snake skeletons are delicate constructions made of small bones linked in long chains, and after death, those chains scatter easily into mud, rivers, or open ground. Entire millions of years pass in near silence within the fossil record, leaving paleontologists to reconstruct ancient animals from isolated vertebrae or partial skulls preserved by extraordinary luck. Into those gaps, genetics has entered like a dim lantern carried through darkness. Scientists now compare the DNA of living species through a field called molecular phylogenetics, tracing tiny genetic mutations that accumulate generation after generation. Over immense spans of time, those changes function almost like a clock. By measuring the differences between modern snakes and other reptiles, researchers estimate that the first true snakes emerged between 128 and 170 million years ago during the Jurassic and early Cretaceous periods. This places their origin tens of millions of years before the asteroid impact that ended the age of non avian dinosaurs. Even language around snakes carries confusion inherited from centuries of fear and fascination. Venom and poison are often treated as the same thing, though biology separates them clearly. Poison harms when touched or eaten. Venom must be delivered directly into tissue, usually through fangs, spines, or stingers. The distinction matters because snakes evolved one of the most sophisticated venom delivery systems in the natural world, though many species remain entirely non venomous. What emerges from all this evidence is not a clean answer, but a narrowing circle around a mystery. Some researchers believe the earliest snakes evolved underground, slipping through soil where legs became cumbersome. Others believe the transformation began near ancient coastlines and shallow seas, where elongated bodies moved efficiently through water. Both theories rest on real fossils, real anatomy, and real geological evidence. The origin of snakes remains unresolved, suspended between stone and genetics, between buried skeletons and living DNA, while the animals themselves continue moving quietly through a world that still carries traces of their beginning. Rain fell differently on the Earth of 150 million years ago. It gathered in warm air that drifted across enormous continents, then settled into forests that seemed to breathe beneath the weight of constant heat. Much of the planet belonged to Gondwana, a southern supercontinent, still connected in vast stretches, though the crust beneath it had already begun to pull apart with almost invisible patience. Shorelines shifted slowly across geological time, and shallow inland seas flooded regions that would one day become deserts and mountains. There were no polar ice caps, no frozen silence at the ends of the world. Global temperatures remained high enough that dense vegetation spread far beyond the tropical zones familiar today. Ferns, cycads, and towering conifers formed layered canopies over wetlands and river plains where humidity lingered close to the ground. Insects filled the air continuously. Amphibians waded in muddy water beneath roots thick as walls. Everything alive existed inside competition that rarely paused. Across this landscape moved dinosaurs in forms so varied they reshaped the structure of entire ecosystems. Sauropods crossed floodplains on pillar like legs, their necks rising above the trees like slow moving cranes. Armored ankylosaurs pushed through undergrowth, carrying entire fortresses across their backs. Early tyrannosaurs had not yet reached the colossal scale they would later achieve, though even then small reptiles lived carefully beneath their shadows. The ground itself trembled often enough that vibration became part of the environment, another current flowing through daily life. Yet the future of snakes was not unfolding among giants. It was taking shape lower down, near the dark spaces beneath roots, stones, and fallen vegetation, where survival favored animals small enough to disappear. The forest floor held a different kind of danger. Predators hunted from above, below, and within the undergrowth itself. Small reptiles faced constant pressure from carnivorous dinosaurs, early mammals, amphibians, and larger lizards competing for the same shelter and prey. In such crowded environments, evolution rarely rewards perfection. It rewards usefulness. Tiny variations appear by accident through mutation and inheritance, then remain only if they help an animal survive long enough to reproduce. Over immense spans of time, bodies drift toward overlooked opportunities. A slightly narrower torso allows easier movement through dense roots. Shorter limbs catch less against packed soil. Flexible ribs permit entry into confined spaces, inaccessible to larger hunters. Gradually, what begins as variation becomes specialization. The ancestors of snakes were moving toward a niche that most legged animals struggled to exploit. Beneath the visible world existed networks of tight tunnels, decaying vegetation, narrow crevices, and compressed leaf litter where long bodies could travel efficiently, but broad limbs became obstacles. In these hidden corridors, speed mattered less than access. Protection came not from armor or size, but from entering spaces where predators could not follow. The transformation did not happen with intention. No ancient reptile chose a future without legs. Instead, generation after generation passed through landscapes that quietly filtered bodies according to what the environment allowed. The Earth of the Jurassic period was not guiding evolution towards snakes specifically. It was creating conditions where certain forms survived more often than others, until one lineage began slipping steadily away from the familiar architecture of lizards and into something the planet had never seen before. Somewhere beneath layers of vanished soil and broken stone, there once moved a reptile small enough to rest in the curve of a human forearm. It slipped through low vegetation and narrow shelters, with a body already beginning to depart from the familiar proportions of ordinary lizards. Paleontologists cannot point to a complete skeleton and declare with certainty that this was the first proto-snake. Instead, the image emerges gradually from fossils, genetics, and anatomical traces carried invisibly inside living species. Piece by piece, a silhouette appears through deep time. This animal was likely between 30 and 60 centimeters long. Its torso had already stretched far beyond the proportions expected for a reptile of its size, giving it a flowing shape that distributed movement across the entire spine rather than concentrating force in the limbs. The legs remained present, though reduced, shorter and less powerful than those of most lizards surrounding it. They still touched the ground, still carried muscle and bone, but the body itself had begun taking over tasks that legs once dominated. Movement through confined terrain favored flexibility over stride. The vertebral column lengthened gradually across generations, adding more joints, more points of articulation, more opportunities for smooth compression against the earth. In places where roots twisted through sediment and narrow cavities folded beneath debris, a long body could travel with unusual efficiency. Limbs increasingly became interruptions along the animal's outline, catching against surfaces that the torso itself could already navigate cleanly. The skull was changing as well, though in quieter ways, hidden beneath skin and muscle. Early proto-snakes still possessed the rigid facial structure typical of reptiles, yet certain bones had begun loosening their ancient connections. Tiny shifts in articulation allowed subtle flexibility around the mouth and jaw. These changes would one day culminate in the extraordinary feeding mechanics of modern snakes, though here they existed only in early form, almost unnoticeable without careful study of fossil anatomy. Unlike living snakes, this ancestor almost certainly blinked. Functional eyelids still covered the eyes, protecting them from soil particles and debris. Modern snakes lost those moving lids long ago, replacing them with transparent scales fused permanently over the eye surface. In the proto-snake, however, the older architecture remained intact, a reminder that evolutionary transitions rarely happen all at once. Ancient traits and emerging ones often coexist together for immense spans of time. The tongue had also begun its transformation into a sensory instrument. Evidence suggests that a forked structure was already present or beginning to develop, paired with the Jacobson's organ embedded within the roof of the mouth. As the tongue moved through air or across surfaces, Chemical particles gathered on its tips and transferred inward, allowing the animal to interpret invisible traces left behind by prey, predators, and its surroundings. The world was becoming less visual and more chemical, rid through drifting molecules rather than distant shapes. This proto-snake spent much of its life close to the ground and likely inside sheltered spaces where compression, flexibility, and subtle movement offered advantages larger reptiles could not easily access. It was neither fully snake nor fully lizard, but something balanced between body plans, carrying remnants of an older anatomy while slowly assembling a new one. Every modern cobra, python, viper, and sea snake descends from creatures shaped much like this, small reptiles moving quietly through hidden spaces, while evolution altered them bone by bone across millions of years. In a slab of pale stone from northeastern Brazil, a body lay curled so delicately that even after more than 100 million years, its outline still seemed almost soft. The fossil was small enough to fit across two human hands, only about 20 centimeters long, yet within that narrow skeleton rested a possibility that unsettled decades of scientific assumptions. When researchers formally described it in 2015 in the journal Science, the animal received a name that immediately carried enormous weight: Tetrapodophis ampilectus, the four-footed snake that embraces. The rocks preserving the specimen dated to the early Cretaceous period, between 110 and 120 million years ago, during an age when flowering plants were only beginning to spread across the planet. Most fossils from this time survive in fragments, isolated vertebrae, or crushed impressions buried in sediment. Tetrapodophis was different. Much of the skeleton remained intact, arranged in remarkable detail along the stone surface. Vertebrae extended in long succession down the body. Tiny ribs curved outward in dense repetition. The skull carried proportions that many paleontologists immediately recognized as deeply snake-like. Then there were the limbs, four small appendages projected from the elongated body, unmistakably anatomical and complete enough to identify individual bones. The forelimbs were extremely reduced, so short they could not realistically support walking. The hind limbs were slightly larger, though still far too delicate for ordinary locomotion. These were not the strong legs of active lizards crossing open terrain. They appeared diminished, transitional structures already drifting away from their original purpose. Researchers studying the fossil proposed that the limbs may have served entirely different functions by this stage of evolution. The tiny forearms could perhaps grasp prey during feeding or help maintain contact during mating. The body itself had likely become the primary engine of movement, while the legs remained only as fading remnants of an older architecture not yet fully erased. Other features strengthened the argument that Tetrapodophis belonged near the base of snake evolution rather than among unrelated reptiles. The torso was unusually elongated. The tail was comparatively short. Certain aspects of the jaw and skull hinted at increasing flexibility, and the arrangement of scales along the body resembled patterns associated with early snakes rather than conventional lizards. To many scientists, the fossil appeared to capture evolution in motion, a reptile suspended midway through the gradual disappearance of limbs. Yet the stone never settled the debate cleanly. In 2021, another analysis challenged the original interpretation entirely. Some researchers argued that Tetrapodophis was not a snake ancestor at all, but instead a different type of elongated lizard converging on a similar body shape through separate evolutionary pressures. They questioned aspects of the skull anatomy and the fossil's uncertain collection history, suggesting the evidence linking it to snakes remained incomplete. The disagreement continues quietly through journals, conferences, and anatomical comparisons measured bone by bone. That uncertainty has become part of the fossil's significance. Paleontology often appears from a distance, like a science of firm conclusions carved directly from rock. Yet discoveries like Tetrapodophis reveal something gentler and more tentative. Ancient life rarely leaves perfect records behind. Sometimes an entire chapter of evolutionary history rests upon a creature no longer than a pencil, preserved in stone while scientists circle carefully around its meaning for years without arriving at complete agreement. Wind moves differently across Patagonia. It travels low over dry plateaus and exposed stone, carrying dust through landscapes that seem almost stripped down to geology itself. In these ancient sediments of Argentina, paleontologists uncovered fossils that clarified one of the most uncertain chapters in snake evolution. The animal was named Najash rhinograena, and unlike several earlier discoveries surrounded by debate, this creature entered the scientific record with unusual certainty. Najash was a true snake. The first fossils were described in 2006, then expanded dramatically by new material published in 2019. Together, the specimens revealed an animal that lived roughly 95 to 100 million years ago during the late Cretaceous period. By then, snakes had already begun diverging far from their reptilian ancestors, yet Najash still carried unmistakable reminders of an older anatomy deep within its body. It was relatively large for an early snake, stretching beyond 1 meter in length with a body built less like a modern burrower and more like an active terrestrial predator. The spine extended in long succession through the torso, already displaying the elongated proportions associated with snakes today. But near the rear of the skeleton, something extraordinary remained attached to the vertebral column. Najash possessed fully formed hind limbs. These were not tiny external spurs hidden beneath muscle, like those seen in living pythons and boas. The legs of Najash were substantial enough to contain recognizable bones extending into small feet. Most importantly, they connected directly to a sacrum, the fused vertebrae structure that anchors hind limbs to the pelvis in four-limbed vertebrates. Modern snakes completely lack this attachment point. In Najash, it still existed, preserved in stone after nearly 100 million years. At the same time, the front limbs had already disappeared entirely. No trace of forelegs remained anywhere in the skeleton. This asymmetry became one of the most important revelations provided by the fossil. Snake evolution had not erased all limbs simultaneously. The front legs vanished first, while the hind limbs persisted for millions of years afterward, lingering through generations as the body continued adapting toward full limblessness. The skull uncovered in the later discoveries added another layer to the picture. Earlier reconstructions imagined primitive snakes with relatively stiff heads compared to modern species, but Najash suggested otherwise. Its skull bones displayed a degree of flexibility and mobility more advanced than scientists had expected for such an ancient snake. Even at this stage of evolution, the feeding apparatus was already shifting toward the kinetic architecture that would later allow snakes to consume prey far wider than their own heads. Equally important was the environment where Najash lived. Geological evidence showed clearly that it inhabited dry terrestrial landscapes rather than marine habitats. This detail carried enormous weight because several other early snake fossils had been recovered from ancient seabeds, leading some researchers to propose that snakes evolved primarily in aquatic settings. Najash complicated that interpretation. Here was an unmistakable early snake with hind legs living firmly on land. Its name carried symbolism as old as storytelling itself. Najash comes from the Hebrew word for the serpent in the Garden of Eden, chosen deliberately by the researchers who described the fossil. Across human history, snakes have occupied mythology, religion, medicine, and fear with unusual persistence. Buried within Patagonian rock, this ancient animal quietly joined that long cultural shadow, not as metaphor or legend, but as anatomy preserved across geological time. Scientific debates rarely resemble arguments in the ordinary sense. More often, they unfold slowly across decades through fossil layers, anatomical comparisons, and quiet revisions inside journal pages read by only a handful of specialists. The origin of snakes remains one of those lingering debates, unresolved not because evidence is absent, but because the evidence points in more than one direction at once. One possibility begins beneath the ground. According to the fossorial hypothesis, the earliest snakes evolved as burrowing reptiles moving through packed soil, loose sediment, and dense leaf litter. In confined underground spaces, long, narrow bodies could travel more efficiently than broad limbed ones. Legs, once useful for climbing or sprinting, became liabilities that caught against surrounding material. Over immense spans of time, natural selection favored bodies capable of compression and smooth forward motion through narrow tunnels where larger animals could not easily follow. Modern snake anatomy preserves several features consistent with this idea. Most snakes lack external ear openings, reducing vulnerable gaps along the skull. Many burrowing species possess extremely reduced eyes, protected beneath scales or thickened tissue. Their skulls are compact and reinforced, able to tolerate pressure from surrounding earth during underground movement. The oldest surviving snake lineages alive today, including blind snakes and thread snakes, spend much of their existence below the surface, moving almost invisibly through soil and decaying vegetation. Yet another possibility drifts through ancient water. The aquatic hypothesis proposes that early snakes evolved in marine or coastal environments where elongated bodies became advantageous for swimming and underwater hunting. Limbs can create drag in water, while long, flexible torsos move efficiently through currents using lateral undulation. Supporters of this theory point towards several important fossils discovered in marine sediments dating back roughly 95 million years. Among the most discussed is Pachyrhacus problematicus, uncovered in Israel within limestone deposits formed beneath shallow seas. The animal possessed a distinctly elongated body, along with small but recognizable hind limbs. Nearby fossils of Haseophis and related species emerged from similar environments connected to ancient coastlines. These discoveries suggested that some early snakes were spending substantial portions of their lives in marine habitats while still retaining remnants of legs. The difficulty is that neither theory explains everything cleanly. The aquatic model struggles against the evidence provided by Najash, whose skeleton and surrounding geology firmly place it on land rather than in water. Its anatomy showed a terrestrial animal moving across dry environments while still retaining hind limbs. At the same time, the burrowing hypothesis faces complications of its own. Certain early snake skulls appear adapted for capturing relatively large prey through flexible jaw mechanics, a feeding strategy difficult to reconcile with a life spent almost entirely underground in cramped tunnels. As a result, the debate continues with unusual persistence. Some researchers suggest snakes may have passed through multiple ecological stages, beginning in one environment before expanding into another. Others argue that separate lineages developed similar features independently, complicating attempts to reconstruct a single, clean origin story. Current scientific consensus leans cautiously toward a fossorial beginning, largely because many anatomical traits align naturally with underground movement. Still, the marine fossils remain embedded in the record like unresolved echoes from ancient coastlines. The earliest history of snakes exists partly in stone and partly in uncertainty, where each newly discovered vertebra or skull fragment has the potential to shift the balance again with only a few centimeters of bone. Inside every vertebrate embryo, long before scales or feathers appear, the body begins as a quiet arrangement of chemical instructions unfolding in sequence. Cells divide, migrate, and specialize according to signals written deep within DNA, following patterns refined across hundreds of millions of years. Legs do not emerge from a single leg gene hidden somewhere in the genome. Instead, they arise from networks of regulatory genes, switching on at precise moments and locations, like lanterns illuminating one chamber after another inside a dark structure. Among the most important of these are the Hox genes, ancient developmental regulators shared across nearly all complex animals. These genes help organize the body from head to tail, determining where ribs form, where vertebrae change shape, and where limbs emerge from the torso. The same underlying genetic architecture builds remarkably different structures across vertebrate life. A human arm, a lizard leg, a whale flipper, and a bat wing all begin through variations of the same developmental machinery, modified over evolutionary time into radically different forms. Snakes still carry this machinery inside their genomes. The genes responsible for limb development were never deleted or erased. They remain present, inherited from four-limbed ancestors that walked the earth long before snakes existed. What changed instead was the regulation surrounding those genes, the timing and intensity with which certain instructions activated during embryonic growth. In 2016, researchers identified one of the most important pieces of this puzzle, a stretch of non-coding DNA known as the ZRS enhancer. Unlike conventional genes that produce proteins directly, enhancers function more like switches controlling when nearby genes activate. In most vertebrates, the ZRS enhancer helps initiate the developmental cascade that produces limbs. In snakes, however, this enhancer contains mutations accumulated gradually across evolutionary time, altering its ability to trigger normal leg formation. The distinction mattered enormously. Evolution had not removed the blueprint for limbs. It had altered the instructions, telling the embryo when to use that blueprint. To test this directly, scientists inserted the snake version of the enhancer into mouse embryos in place of the ordinary mammalian sequence. The resulting mice developed severely reduced limbs, confirming that tiny changes within a regulatory region could profoundly reshape the body plan of an animal. A few altered molecular signals, accumulated through millions of generations, had redirected the anatomy of an entire lineage. The loss of legs carried substantial consequences. Limbs in vertebrates serve as anchors for major muscle groups and provide efficient leverage for rapid movement across land. By abandoning them, early snakes surrendered the standard locomotive strategy used by nearly every large terrestrial vertebrate. They also lost the ability to grasp, climb in the conventional reptilian sense, or manipulate the environment with appendages. Yet evolution rarely preserves costly changes unless another advantage compensates for them. As the limbs diminished, the rest of the body reorganized around a different approach to movement and survival. Musculature redistributed along the torso, the spine elongated and diversified. New forms of locomotion emerged from hundreds of articulating vertebrae, working in coordinated sequence. Even now, traces of the older body plan remain hidden inside every developing snake embryo. For a brief period during early growth, tiny limb buds still appear along the body before fading again under the influence of altered regulatory signals. Deep within modern snakes, the memory of legs has never completely vanished. It persists quietly in dormant instructions carried from a distant ancestry that the genome still remembers. A human spine feels rigid because it is built around stability. The vertebrae support upright posture, protect the spinal cord, and anchor muscles that move arms and legs in coordinated patterns. In snakes, the spine followed an entirely different path through evolution. Instead of becoming a framework for limbs, it became the primary structure through which nearly every interaction with the world occurs. The body itself transformed into a continuous, muscular instrument capable of astonishing precision. Most mammals possess somewhere between 7 and 25 vertebrae. A snake may carry anywhere from 100 to more than 400 depending on species. The reticulated python, the longest living snake on Earth, contains roughly 300 vertebrae linked together in an extended chain. Certain elongated species exceed even that number, producing bodies where movement is distributed across hundreds of independent articulations rather than concentrated into a few major joints. Each vertebra connects to surrounding muscle through an intricate lattice of tendons and ribs. As waves of contraction travel along the torso, every segment contributes a tiny adjustment in angle and pressure. The result is locomotion that appears fluid from a distance, though beneath the skin it consists of countless coordinated mechanical actions unfolding simultaneously. No-legged vertebrate moves this way because no-legged vertebrate possesses such a densely segmented body. This architecture allows snakes to employ several distinct methods of locomotion depending on terrain. The most familiar is lateral undulation, where curves pass rhythmically down the body while scales push against irregularities in the environment. Through grass, shallow water, or uneven ground, the snake advances by converting sideways force into forward motion. Other methods are stranger still. Rectilinear movement allows large snakes, such as boas and pythons, to crawl forward in nearly straight lines using alternating rows of belly scales. The body advances gradually, almost imperceptibly, producing the quiet, gliding motion often associated with large constrictors. Sidewinding appears in desert species, crossing loose sand, where sections of the body lift and reposition in rolling loops that minimize sinking into unstable surfaces. Concertina locomotion dominates inside tunnels or narrow passages, with alternating portions of the body gripping and extending like a slowly compressing spring. No other vertebrate combines this many locomotive systems within a single body plan. The spine itself became adaptable enough to solve entirely different environmental problems without requiring separate anatomical structures. The elongated torso altered internal anatomy as profoundly as external movement. In mammals, most major organs cluster compactly within the rib cage and abdomen. Snakes instead arrange their organs sequentially down the length of the body. One lung may shrink dramatically while the other elongates. The liver, stomach, intestines, and reproductive organs stretch along the torso like components distributed through a narrow corridor. This arrangement permits feeding events that seem disproportionate to the animal itself. A Burmese python can consume prey as large as a deer, then remain motionless for days or weeks while digestion unfolds. During this process, the body enters an intense metabolic state. The heart enlarges temporarily, digestive tissues increase in mass, enzyme production accelerates. Once digestion finishes, many of those organs reduce again, conserving energy until the next meal. That efficiency defines much of snake biology. Some species survive on only a handful of meals each year, maintaining themselves through remarkably low metabolic demands. The same body that surrendered limbs eventually became one of the most energy efficient vertebrate designs ever produced, capable of crossing deserts, forests, rivers, and tunnels using little more than muscle, bone, and time. A snake feeding can appear almost unreal in slow motion. The mouth opens far beyond proportions that seem mechanically possible. Skin stretches around prey wider than the head itself, and the body advances steadily over the meal, as though the animal were pulling itself across the surface of another creature. For centuries, this led to a common explanation that snakes dislocate their jaws while feeding. In truth, the jaw was never fixed into a rigid position to begin with. The skull of a snake is kinetic, built from bones connected through flexible ligaments rather than tightly fused joints. Instead of functioning as a single solid structure, different parts of the skull move semi-independently during feeding. This arrangement allows the entire head to expand, shift, and reposition itself around prey with remarkable control. The architecture did not emerge suddenly. Fossils suggest these modifications were developing gradually during the same evolutionary period when limbs were disappearing and the body itself was becoming increasingly elongated. The lower jaw reveals this flexibility most clearly. In mammals, the left and right halves of the jaw are fused into a single solid unit. In snakes, the two halves remain separate, joined only by an elastic ligament near the front of the mouth. This permits each side to move independently during swallowing. One side grips while the other advances forward, creating an alternating sequence that slowly pulls prey deeper into the throat. The teeth contribute to this process with equal specialization. Most snake teeth curve backward toward the throat, like rows of tiny hooks. Once prey enters the mouth, escape becomes extremely difficult because every movement backward catches against the inward-facing teeth. Rather than chewing or tearing food apart, the snake uses coordinated jaw movements to ratchet the prey inward a little at a time. A crucial element in this mechanism is the quadrate bone, a long, movable structure connecting the lower jaw to the skull. In many vertebrates, this region remains compact and relatively fixed. In snakes, the quadrate extends outward and pivots freely, functioning almost like an additional lever that dramatically increases how wide the mouth can open. The gape becomes less dependent on skull width and more dependent on flexibility distributed across multiple moving components. Feeding places unusual demands on breathing as well. During the swallowing process, prey may occupy nearly the entire mouth and throat for extended periods. To compensate, snakes can extend the trachea forward beneath the meal, allowing air to continue flowing into the lungs while the rest of the head remains occupied. A large feeding event may continue for hours, especially in constrictors consuming mammals or birds significantly wider than the snake's own body. What makes this anatomy particularly remarkable is that many of its defining features appeared before snake evolution fully completed the transition into limblessness. Early snake fossils from roughly 100 million years ago already display increasing skull flexibility alongside diminishing hind limbs. The feeding system and the elongated body were evolving together, each influencing the survival value of the other across immense stretches of time. A body capable of consuming unusually large prey gained access to meals unavailable to most predators of similar size. The kinetic skull transformed limitations into opportunities. Without claws to grasp or limbs to manipulate food, snakes evolved an entirely different solution, one based not on force or tearing, but on patience, flexibility, and the gradual mechanical precision of moving bones working together beneath stretched skin and scales. Night settles differently for a snake than it does for most animals. Darkness does not erase the world around it or reduce the landscape to silence. The ground continues speaking through vibration. The air remains dense with chemical traces, and warm bodies still radiate heat into the surrounding darkness like faint, invisible lanterns. A snake moves through this hidden layer of information continuously, reading signals that most vertebrates never evolved to notice. Unlike mammals, snakes have no external ear openings. Airborne sounds that humans perceive as conversation, music, or distant movement barely register in the same way. Instead, much of their hearing arrives through direct physical contact with the environment. Vibrations traveling through soil, stone, or wood pass into the lower jaw, then transfer through specialized bones toward the inner ear. The snake effectively listens through pressure transmitted across the earth itself, detecting movement through surfaces before shapes become visible. This relationship with the ground reshaped the sensory priorities of the entire lineage. Vision remained useful in many species, especially arboreal and daytime hunters, yet the snake nervous system increasingly emphasized other forms of perception operating beyond ordinary sight. Among the most remarkable of these is the forked tongue, an organ so familiar in appearance that its actual purpose often goes unnoticed. The tongue is not a threat display and not a weapon. It functions as a highly refined chemical sampling device. With each flick, microscopic particles suspended in air or resting on surfaces adhere to the moist tips of the tongue. Because the tongue divides into two separate forks, samples are collected simultaneously from slightly different locations in space. When the tongue retracts, each tip enters a corresponding chamber of the Jacobson's organ located in the roof of the mouth. The brain compares the intensity of chemical signals arriving from both sides, producing something similar to directional smell. A stronger concentration on one fork reveals where a scent trail grows more intense. Through repeated sampling, the snake constructs a moving chemical map of its surroundings, following prey, identifying predators, or locating potential mates with remarkable precision. The process resembles stereo hearing or binocular vision, except built entirely from drifting molecules suspended in the environment. Some snake lineages carried sensory specialization even further. Pit vipers, pythons, and certain boas evolved facial pit organs positioned between the eye and nostril. These structures detect infrared radiation emitted by warm objects. Heat itself becomes perceptible. The sensitivity of these organs is extraordinary. Certain species can distinguish temperature differences as small as 0.003 degrees Celsius. In total darkness, a python may strike a warm-blooded animal with accuracy comparable to feeding in daylight because the prey remains visible through its body heat alone. The pits contain no lenses and no retina like ordinary eyes, yet the signals travel into regions of the brain associated with visual processing. The result is a layered sensory experience where thermal information overlays conventional sight. Chemical traces, substrate vibrations, and infrared radiation merge continuously inside the snake nervous system, forming a perception of the world fundamentally unlike human experience. The loss of limbs altered far more than movement. It redirected evolutionary pressure towards sensory systems capable of interpreting environments through texture, chemistry, and heat rather than relying primarily on vision or sound. In doing so, snakes became among the most comprehensively equipped predators ever produced by vertebrate evolution, navigating darkness and hidden spaces with senses tuned to signals that remain entirely invisible to us. Long before humans learned to distill medicines from plants or shape metal into surgical tools, snakes had already begun conducting chemistry inside their own mouths. Deep within specialized glands behind the eyes, proteins and enzymes accumulated in carefully balanced mixtures capable of altering blood pressure, destroying tissue, interrupting nerve signals, or stopping the rhythm of a heart. Venom was not a single invention appearing once in evolutionary history. Molecular evidence now suggests it emerged independently, at least twice among snakes, possibly more, through separate lineages, refining similar biological strategies. At its origin, venom began as something far more ordinary. The glands producing it evolved from ancestral salivary glands already present in vertebrates long before snakes appeared. Across millions of years, natural selection gradually favored saliva components that weakened prey or assisted digestion. Small biochemical advantages accumulated generation after generation until saliva transformed into a weapon carried directly inside the mouth. Venom itself leaves no fossil imprint. Proteins decay quickly after death, vanishing long before skeletons enter stone. Scientists instead infer the appearance of venom through skull anatomy, fang structure, and gland placement preserved in fossilized remains. The earliest strongly venomous snakes likely emerged around 60 million years ago, after the extinction of the non-avian dinosaurs had already reshaped ecosystems worldwide. Inside a venom gland, chemistry becomes extraordinarily complex. A single species may produce between 20 and more than 100 distinct molecular compounds, each targeting different biological systems. Some toxins attack the nervous system directly, preventing communication between nerves and muscles until breathing stops. These neurotoxins act with frightening speed in certain species, shutting down movement while prey remains fully conscious. Other venoms specialize in destroying blood vessels, dissolving tissue, or disrupting clotting mechanisms. Hemotoxins damage circulation and surrounding flesh, while cytotoxins rupture cell membranes themselves. Different snake lineages arrived at these chemical solutions independently. Elapids, such as cobras and taipans, evolved venom systems dominated largely by neurotoxins, while many vipers refined venoms causing catastrophic tissue destruction and internal bleeding. Delivery mechanisms diversified as well. Some snakes developed long, hollow front fangs capable of injecting venom deeply with remarkable precision. Others retained rear-positioned fangs connected to grooved channels carrying venom gradually into bite wounds. A few species deliver toxins through modified teeth without fully enclosed fangs at all. Among modern snakes, the inland taipan of Australia produces the most toxic venom known from any land species. A single bite contains enough venom to kill roughly 100 adult humans or hundreds of thousands of mice under laboratory measurements. Yet, despite this potency, the inland taipan has never caused a recorded human death. It inhabits remote inland regions and behaves with unusual reluctance toward aggression, avoiding confrontation whenever possible. Venom transformed snake ecology profoundly because it expanded access to prey impossible to overpower through strength alone. A relatively slender predator could now subdue mammals, birds, reptiles, or amphibians much larger than itself through chemistry rather than physical force. The body no longer needed claws or grasping limbs when proteins carried the work inward through a pair of fangs. Even behavior shifted alongside these biochemical changes. king cobras, the largest venomous snakes alive today, construct nests from gathered vegetation and remain nearby, guarding their eggs after laying them. No other snake species shows this level of parental investment. Some researchers suspect the enormous metabolic cost of producing complex venom may have influenced the evolution of such protective behavior, linking chemistry, energy, and reproduction together within the long, unfolding story of snake evolution. Most ancient animals leave behind fragments of themselves eventually. Teeth settle into riverbeds, shells harden beneath sediment, massive bones survive compressed beneath layers of stone for millions of years. Snakes rarely grant paleontology such generosity. Their skeletons are delicate constructions made from hundreds of narrow bones linked in long, flexible chains, and after death, those chains separate quickly. A carcass exposed to water currents, scavengers, weather, or shifting soil may scatter completely within days, leaving almost nothing stable enough to fossilize. For this reason, the fossil history of snakes remains unusually sparse compared to other major vertebrate groups. Nearly 3,900 snake species exist today across deserts, forests, oceans, mountains, and wetlands, yet only a small fraction possess meaningful fossil ancestors identified with confidence. Entire branches of snake evolution vanish into geological silence for tens of millions of years at a time. Even under ideal conditions, preservation requires improbable circumstances. A snake must die in an environment where decomposition slows rapidly, burial occurs gently, and surrounding sediment remains undisturbed long enough for minerals to replace organic tissue. Most never experience such conditions. They disappear back into the landscape almost immediately, leaving evolutionary history incomplete before it can enter the rock record. When discoveries do occur, they demand extraordinary patience. Significant snake fossils are rarely uncovered during a single dramatic excavation. More often, they emerge through years of fieldwork, followed by equally slow laboratory preparation. Paleontologists may spend months removing stone grain by grain beneath microscopes, exposing vertebrae thinner than matchsticks without damaging them. Once prepared, the fossils pass through additional years of anatomical analysis, imaging, comparison with living species, and peer review before publication finally enters the scientific literature. The fossils of Najash from Patagonia illustrate this timescale clearly. Specimens were collected across multiple expeditions between 2006 and 2019, representing 13 years of work before researchers assembled the complete anatomical picture that reshaped understanding of early snake evolution. In paleontology, a single scientific paper may embody decades of excavation, transport, preparation, and comparison hidden quietly behind a few pages of published text. Because the record is so incomplete, every major discovery carries disproportionate weight. One newly uncovered skull or partial spine can alter estimates for when snakes originated, whether early forms lived primarily on land or near water, and how rapidly key anatomical traits evolved. Entire theories sometimes shift around a specimen small enough to fit inside a drawer. There is also the unsettling possibility that important fossils have already been found without being recognized. Museum collections across the world contain countless isolated vertebrae and reptile fragments gathered over centuries of fieldwork. Some may belong to early snakes, misidentified as ordinary lizards, and filed away long before modern evolutionary frameworks existed. Hidden discoveries may already sit quietly inside cabinets, waiting for someone to notice a subtle anatomical detail overlooked decades earlier. In response to these gaps, scientists increasingly rely on genetic evidence from living species to reconstruct evolutionary relationships. Molecular analysis can reveal patterns impossible to detect through fossils alone, tracing divergence times through accumulated mutations preserved within DNA. Yet even these methods carry uncertainty, because they must ultimately anchor themselves against the fragmentary fossil record available. That incompleteness has shaped snake paleontology into an unusually cautious science. Researchers often speak in probabilities rather than certainties, acknowledging openly where evidence ends and interpretation begins. The gaps themselves have become part of the story, reminders that deep time rarely preserves a perfect archive, especially for creatures built from bones delicate enough to vanish almost without a trace. For a brief period in Earth's history, nearly every ecosystem on the planet began to fail at once. Forests burned across continents, oceans acidified, sunlight faded behind dense clouds of pulverized rock and soot drifting through the atmosphere. Food chains that had remained stable for millions of years collapsed with astonishing speed, and countless species disappeared into extinction before the world had time to recover. 66 million years ago, an asteroid estimated between 10 and 15 kilometers wide struck the Yucatan Peninsula in what is now Mexico. The impact released energy equivalent to billions of nuclear weapons, excavating enormous volumes of debris and igniting global wildfires that spread across vast regions of the planet. Dust and aerosols rose high into the atmosphere, where they blocked sunlight for months, possibly years, reducing photosynthesis across land and sea. The extinction that followed erased roughly 75% of species on Earth. Non-avian dinosaurs vanished completely, along with marine reptiles, ammonites, and innumerable smaller organisms woven into ancient ecological networks. Yet, among the survivors moving quietly through this ruined world were snakes. Their survival may have depended less on strength than on restraint. Unlike mammals and birds, snakes are ectothermic. Their bodies do not generate large amounts of internal heat continuously, which means their energy demands remain comparatively low. In stable environments, this can appear like a limitation. During catastrophe, it became an advantage. As temperatures dropped and food sources disappeared, many warm-blooded animals could not sustain the metabolic cost of remaining alive. They required constant calories simply to maintain body temperature. Snakes required far less. Many species could retreat underground, sheltering inside burrows, crevices, fallen debris, or sediment where temperatures fluctuated less severely than on the surface. Their metabolism slowed dramatically. Heart rate decreased, digestion halted, activity nearly ceased. Some modern snakes can already survive months without feeding under ordinary conditions, and their ancestors likely possessed similar physiological resilience during the aftermath of the impact. The post-impact environment also favored opportunistic feeding. Carcasses littered many ecosystems, while populations of small surviving vertebrates and invertebrates fluctuated unpredictably through damaged habitats. Snakes were well suited to exploit these unstable food sources because their diets were flexible. Different species consumed rodents, amphibians, eggs, insects, carrion, fish, or other reptiles depending on availability. Survival did not depend on a single specialized prey species remaining intact. As ecosystems slowly stabilized across the following millions of years, new ecological spaces opened almost everywhere. Large dominant predators had vanished, forests regenerated under altered climates, and food webs reorganized themselves around surviving lineages. Snakes expanded rapidly into these emptied niches with extraordinary evolutionary speed. Within roughly 10 million years after the asteroid impact, many major snake lineages recognizable today had already begun to diversify. Some adapted toward aquatic environments, others specialized for trees, deserts, wetlands, or underground habitats. Venom systems became more sophisticated, body sizes diversified dramatically. The extinction event that ended the age of dinosaurs became, for snakes, a period of expansion. There is something quiet about that survival. Snakes did not endure through armor, intelligence, or physical dominance over the catastrophe itself. They endured through flexibility, low energy demands, patience, and the capacity to remain hidden while the planet reorganized around them. When conditions finally improved, they emerged into a world suddenly filled with ecological opportunities left vacant by extinction. The world left behind after the asteroid impact was strangely open. Forests regrew across altered continents. Rivers shifted course through landscapes no longer dominated by giant reptiles. Predators that had controlled ecosystems for millions of years were gone, and in the quiet ecological space that followed, snakes began spreading into roles they had never occupied before. At the time of the extinction event, only a small number of snake lineages likely remained alive. Today, that handful has become nearly 3,900 known species distributed across more than 30 families. The transformation occurred with unusual speed in geological terms, producing one of the clearest examples of adaptive radiation among vertebrates. The body plan that once evolved under narrow evolutionary pressures suddenly encountered a planet filled with opportunity. Snakes entered wetlands, forests, deserts, coastlines, and eventually the open ocean itself. Different lineages adapted independently to conditions separated by thousands of kilometers and entirely different climates, yet the same elongated body repeatedly proved capable of remarkable versatility. Some species returned to water through multiple separate evolutionary events. Marine snakes developed paddle-shaped tails that generated efficient propulsion through coastal currents and coral reefs. Certain sea snakes evolved skin capable of limited oxygen absorption directly from surrounding water, extending the time they could remain submerged. Specialized glands near the tongue began removing excess salt from the bloodstream, allowing these reptiles to survive in marine environments that would dehydrate most terrestrial animals. Elsewhere, tropical forests produced an entirely different set of pressures. Arboreal snakes moved upward into dense canopies where balance and grip mattered more than raw speed. Prehensile tails appeared in several lineages, functioning almost like additional anchoring limbs around branches. Some tree-dwelling species evolved flattened bodies that distributed weight more evenly while climbing through narrow vegetation high above the forest floor. Among the most unusual are the paradise flying snakes of Southeast Asia. When launching from elevated branches, these snakes flatten their ribs outward until the body forms a shallow concave shape. By undulating through the air in controlled waves, they can glide horizontally for distances approaching 10 meters before landing. The movement resembles swimming suspended briefly above the forest below. Deserts shaped snake evolution differently again. Loose sand punished ordinary locomotion, leading certain species toward side-winding movement, where portions of the body lift clear of the surface, while only limited contact points touch the ground. Heat-resistant physiology emerged alongside venom systems increasingly specialized for small mammals dominating arid ecosystems. Some desert snakes became nocturnal almost entirely, moving across cooling dunes beneath darkness while conserving water and energy. The diversification was not random wandering through evolutionary possibility. It followed ecological opportunity with remarkable precision. Whenever a habitat offered food, shelter, or reduced competition, some lineage of snake eventually adapted toward exploiting it. Similar environmental pressures repeatedly produced similar anatomical solutions even among distant relatives. Across 66 million years, the snake body proved capable of extraordinary modification without abandoning its fundamental architecture. Elongation, flexible movement, chemical sensing, and kinetic skull mechanics remained intact while surrounding traits shifted in response to climate and geography. The result was not a single successful species, but thousands, each carrying variations of the same ancient design into entirely different corners of the planet. What began as a narrow lineage, surviving catastrophe gradually unfolded into one of the most geographically widespread and ecologically adaptable groups of predators that Earth has ever produced. Along the lower body of a python, near the cloaca, where the digestive and reproductive systems open to the outside world, there are two small projections that resemble curved claws pressing outward beneath the scales. They are easy to overlook at first glance. Many people handling a python never notice them at all. Yet those tiny structures are among the most direct surviving traces of the moment snakes still carried legs. Pythons and boas occupy unusually ancient positions on the snake evolutionary tree. Among living snake families, they preserve skeletal features that vanished entirely in most later lineages. Buried beneath layers of muscle and scales inside their bodies rests a reduced pelvic girdle along with remnants of hind limb bones once connected to walking legs. The structures are incomplete now, transformed almost beyond recognition by millions of years of reduction. In pythons, the remaining bones include a small femur, the same thigh bone found in mammals, birds, and lizards. What once supported weight against the ground has become a detached fragment floating within surrounding tissue, no longer connected to a functioning leg. Nearby remnants of the pelvis persist as delicate internal structures, detached from the spine and serving none of their original locomotive purpose. Externally, the surviving tips emerge as spurs. In males, these spurs are larger and more pronounced than in females, suggesting that natural selection continues, preserving them for a reason. During courtship and mating, male pythons and boas use the spurs to scratch, tap, and stimulate the female's body as they align themselves for reproduction. Ancient walking structures became tactile reproductive tools instead. This transformation reveals something subtle about evolution. Traits do not always disappear completely when their original purpose vanishes. Sometimes they persist because another function emerges before total reduction occurs. In pythons and boas, the remnants of hind limbs survived not because the animals still needed to walk, but because those structures retained enough reproductive value to avoid being eliminated entirely. Ball pythons, green tree pythons, and reticulated pythons all carry these skeletal echoes. Imaging studies examining their anatomy reveal what looks almost like a faded architectural blueprint hidden beneath the body wall. Bones once anchored firmly to a walking skeleton now drift internally with no contact to the ground and no mechanical role in locomotion. They remain suspended inside the animal like forgotten supports from an earlier design. The persistence of these structures across nearly 100 million years is remarkable in itself. Evolution tends to remove features that consume energy without providing advantage. The fact that vestigial limb remnants still survive suggests they continue offering small but meaningful benefits through mating behavior and reproductive success. They are therefore not vestigial in the popular sense of being useless. Their role simply changed. What once pushed against prehistoric soil now brushes lightly against another snake during courtship. The function narrowed, specialized, and persisted long after walking disappeared entirely. There is something strangely intimate about those hidden bones. Inside a modern python rests physical evidence of an older animal still partially preserved within the newer one, not metaphorically, but literally. The skeleton continues carrying fragments of a body plan inherited from distant ancestors that moved through the world on legs beneath ancient forests while dinosaurs still dominated the land above them. Inside a snake egg, long before scales begin to show their final patterns, the body briefly remembers an older design. Beneath translucent embryonic tissue, tiny protrusions emerge along the lower sides of the developing animal, small rounded structures no larger than grains of dust. Under a microscope, they are unmistakable. They are the beginnings of legs. Every snake embryo studied so far begins development this way. At nearly the same stage, when lizard embryos activate the genetic machinery responsible for forming limbs, snake embryos activate it as well. The process starts normally. Developmental signals move through the growing body. Cells divide and organize into early limb buds. For a short period, the embryo follows instructions inherited from four-limbed ancestors, stretching far back into reptilian history. Then the process stops. Within days, the limb buds stall and begin to regress. The structures shrink rather than expand, fading back into the surrounding tissue before fully formed legs can emerge. In living pythons and boas, this sequence can be observed directly during embryonic development. It is not symbolic reconstruction or evolutionary speculation. The body initiates leg formation, then actively suppresses it before completion. This distinction matters profoundly in developmental biology. Snakes did not lose legs because embryos simply failed to begin constructing them. The genetic program still activates. The interruption occurs later, after the developmental pathway has already started unfolding. Researchers eventually traced much of this suppression to the same regulatory region identified earlier in studies of snake genetics, the ZRS enhancer controlling limb development. In ordinary vertebrates, this enhancer activates genes guiding the growth of limbs outward from the torso. In snakes, accumulated mutations altered the enhancer's behavior, weakening the developmental signals needed to sustain continued limb growth. Developmental biologists studying embryos have identified remarkably precise windows during which this shutdown occurs. Timing is critical. A slight change in when developmental instructions activate or cease can reshape the entire anatomy of an organism. In snakes, the interruption arrives early enough that mature limbs never form, yet late enough for the embryo to reveal traces of its ancestral blueprint before suppression takes hold. Experiments exploring this process produced some of the most striking findings in modern evolutionary biology. In laboratory studies, researchers replaced the mutated snake enhancer with a functional version taken from mice. The altered snake embryos began developing more substantial limb structures than normal before broader developmental incompatibilities eventually halted further growth. The experiments were not attempts to recreate prehistoric snakes. Their purpose was far more fundamental: to understand how tiny regulatory changes can reorganize entire body plans across evolutionary time. Snake embryos became uniquely valuable to science because they preserve a visible transition between ancestral and modern anatomy inside a living developmental process. The evolutionary history remains partially written into the embryo itself, replayed briefly every generation before vanishing again. There is something almost ghost-like about those disappearing limb buds. For a few quiet days, The embryo carries evidence of a body its species has not possessed for tens of millions of years. The instructions survive, dormant but incomplete, embedded deep within the developmental architecture of the animal. Evolution altered the outcome without fully erasing the pathway that once produced walking legs beneath ancient reptilian skin. Motion in a snake does not begin in the way movement begins for most animals. There are no shoulders shifting weight, no hips driving the body forward, no limbs striking against the ground in repeating rhythm. Instead, movement travels through the entire length of the animal at once, carried by hundreds of vertebrae working together in continuous coordination. Beneath the scales, the nervous system responsible for this process is unlike that of almost any other vertebrate alive today. In mammals, birds, and lizards, locomotion is organized around limbs. Different regions of the spinal cord manage separate appendages, while the torso mainly stabilizes the body. A snake could not rely on such a structure. Some species possess more than 400 vertebrae, each connected to muscles capable of independent contraction. Coordinating that many moving parts through direct commands from the brain alone would create impossible delays. Instead, snakes evolved a distributed motor system spread along nearly the entire spinal cord. Neural circuits positioned throughout the body generate repeating waves of muscular activity automatically once movement begins. The brain provides broad control over direction and intensity, but the detailed timing of motion is delegated locally along the spine itself. Every curve of the body influences the next curve in sequence. This arrangement allows snakes to perform several entirely different styles of locomotion. In lateral undulation, muscular waves push against rocks, roots, or uneven ground. In rectilinear movement, the ribs and belly scales lift and pull sections of the body forward with remarkable smoothness. Sidewinding shifts wade across loose sand with minimal slipping, while concertina movement grips narrow tunnels in alternating sections. Each method depends on continuous communication between muscles, vertebrae, and sensory receptors embedded across the body. The spinal cord of a snake contains proportionally more motor neurons per unit length than most vertebrates. Signals move constantly between the body and nervous system as the animal measures pressure, texture, vibration, and resistance from the surrounding environment. A snake pressed against the earth is continuously gathering mechanical information through nearly its entire surface. This distributed neural structure also explains a phenomenon long observed in injured snakes. Coordinated movement can continue briefly even after severe spinal damage or in documented cases after decapitation. Reflex circuits within the spinal cord remain active for a period of time without higher brain control. The body still responds to touch and pressure through local neural pathways. The brain itself reflects the sensory priorities of snake evolution. Compared with similarly sized lizards, snakes devote much larger regions to chemical and vibrational processing while depending less heavily on vision. Signals gathered through the forked tongue and Jacobson's organ feed into enlarged olfactory centers capable of constructing detailed chemical maps of the environment. Pit vipers evolved an additional sensory layer. Infrared detecting organs send thermal information into a specialized midbrain region absent in most other snakes. Heat, vibration, scent, and motion are merged into a unified sensory picture processed continuously in real time. Only in the early 2000s did researchers fully begin mapping these neural pathways. The principles discovered inside the snake nervous system now influence robotics research, especially in machines designed to move through confined or unstable environments with the same flowing adaptability carried through snake evolution for millions of years, long after the asteroid impact had erased the great non-avian dinosaurs. The forests of ancient South America settled into a different kind of dominance. Vast wetlands spread beneath dense tropical canopies. Rivers moved slowly through heat, heavy air, and in the swamps of the Paleocene world, a serpent emerged whose scale still feels difficult to picture against the size of living animals today. Its remains were discovered in the Sarajan coal mine of modern Colombia, preserved in rocks dating to approximately 58 to 60 million years ago. The animal became known as Titanoboa saragenensis, and from the first measurements of its fossil vertebrae, paleontologists understood they were looking at something beyond any snake previously known. The vertebrae themselves were enormous, larger than a human hand, yet unmistakably snake vertebrae in structure. By comparing them with the skeletons of living boas and pythons, researchers estimated a body length approaching 13 meters and a mass exceeding 1,100 kilograms. Even the largest reliably measured reticulated pythons alive today reach only about 7 meters. Titanoboa existed on an entirely different scale. Such immense size was not simply a biological accident, it was tied directly to the climate of the world that produced it. Snakes are ectothermic animals, their metabolism dependent on environmental warmth rather than internally generated body heat. In cooler climates, very large body sizes become increasingly difficult because chemical processes slow as temperatures fall. A giant snake living in the modern world would struggle to sustain the metabolic activity required for movement and digestion. The Paleocene epoch offered conditions unlike those of the present Earth. Global temperatures were estimated to be roughly 6 to 8 degrees Celsius warmer than modern averages. Tropical forests extended through landscapes that remained warm year-round, without polar ice caps cooling the planet. Rivers, swamps, and flooded forests formed stable heat reservoirs where enormous ectothermic predators could survive efficiently. Titanoboa appears to have spent much of its life in water. Its immense bulk would have been easier to support while submerged, and the anatomy of its skull suggests a diet focused largely on large fish. Some researchers also believe it preyed upon early crocodilian relatives sharing the same wetlands. Like modern constrictors, it likely subdued prey through pressure rather than venom, coiling its body around struggling animals beneath dark river surfaces. The discovery of Titanoboa in 2009 transformed more than the study of snakes alone. Paleontologists realized the animal could serve as a biological climate indicator. Because ectothermic body size is constrained by environmental temperature, the dimensions of Titanoboa allowed scientists to estimate the minimum warmth required to sustain such an animal. Those calculations closely matched temperature estimates already derived from fossil plants and chemical analysis of ancient sediments. In this way, the skeleton of a giant snake became evidence for the atmosphere of an extinct world. Its vertebrae carried information not only about anatomy, but about rainfall, heat, and the structure of forests that vanished tens of millions of years ago. The rivers that once carried Titanoboa no longer exist, and the climate that sustained it has long since faded from Earth. Yet, inside fragments of fossil bone, the temperature of that vanished planet still quietly remains. Long before human language emerged, before hands shaped tools, or fire altered the rhythm of the night, another relationship had already been unfolding across forests and riverbanks. Small mammals moved through branches and undergrowth, while silent reptiles occupied the same landscapes beside them. Across millions of years, Those encounters left traces not only in fossil bones, but deep inside the architecture of the primate nervous system. The lineage that would eventually produce humans shared habitats with venomous snakes by the time the first true primates appeared, roughly 55 to 65 million years ago. These early primates were small animals living in dense tropical forests where visibility was limited and survival depended on rapid recognition of danger. A motionless snake, coiled along a branch or hidden beneath leaves, represented a threat requiring immediate detection. In 2006, evolutionary biologist Lynn Isbell proposed what became known as the snake detection theory. The idea suggested that many defining features of primate vision evolved under selective pressure from snakes. Forward facing eyes, enhanced depth perception, improved color discrimination, and unusually rapid visual processing may all have been shaped partly through repeated encounters with venomous reptiles. The theory gained support from neuroscience. Within the primate brain exists a specialized visual pathway running through the pulvinar nucleus of the thalamus, a region that responds with remarkable speed to snake-like shapes. Researchers observed that both humans and non-human primates consistently detect snakes faster than other threatening animals, even when the images appear briefly or are partially concealed within visual clutter. Experiments with wild monkeys revealed similar patterns. Individuals raised without direct exposure to snakes still reacted more quickly to snake forms than to other predators. Human participants showed the same response across cultures and environments. Even infants only a few months old displayed heightened attention, widened pupils, and measurable physiological reactions when presented with images of snakes compared with flowers, fish, or neutral objects. Researchers increasingly concluded that this response is not simple learned fear. Instead, it appears to be a prepared neural bias, a tendency built into the visual system itself through millions of years of evolutionary pressure. Organisms that recognized snakes more efficiently survived encounters more often, passing those perceptual advantages into future generations. The relationship extended beyond biology into symbolism. Human societies, separated by oceans and lacking historical contact, independently filled myths, religions, and artwork with serpents. Snakes appeared as guardians, creators, deceivers, healers, and beings linked to transformation or death. Their forms wound through temple carvings, burial chambers, painted ceramics, and oral traditions on nearly every continent humans inhabited. This widespread symbolic presence may reflect something deeper than cultural coincidence. The snake occupies an unusually privileged position in human attention. The nervous system notices its outline rapidly, tracks its movement instinctively, and stores the image with unusual persistence. Few other animals provoke such immediate cognitive recognition across so many unrelated societies. In this way, the story of snakes and humans became intertwined long before humanity understood evolution or biology. The predator hidden in ancient forests may have influenced the refinement of primate perception itself, shaping the visual precision later used to identify faces, navigate landscapes, interpret symbols, and eventually read written language beneath artificial light many millions of years later. Across forests, deserts, wetlands, and oceans, evolution has repeatedly arrived at the same strange shape. Long bodies close to the ground, limbs reduced or absent, movement flowing through muscular waves instead of footsteps. The snake form did not emerge only once. Nature discovered versions of it, again and again, through separate lineages divided by millions of years of ancestry. One of the clearest examples appears in the amphisbaenians, a group of burrowing reptiles sometimes called worm lizards. They are not snakes, nor are they close snake relatives, yet their bodies tell a remarkably similar story. Their limbs disappeared independently. Their eyes became reduced beneath thickened skin, and their skulls hardened into compact structures capable of forcing through soil. Underground tunnels reward narrow, flexible bodies and punish protruding limbs that snag against confined walls. Elsewhere, another imitation emerged through the slow worm of Europe. At first glance, it appears nearly identical to a small snake moving through grass. The body is elongated, the limbs absent, the movement serpentine and smooth. Yet slow worms are not snakes at all. They are legless lizards, descendants of ancestors that lost their limbs through a completely separate evolutionary pathway. Beneath the skin, traces of their history remain. Vestigial shoulder bones persist inside the body, structures true snakes no longer possess. Even amphibians arrived at similar solutions. Caecilians, which resembled dark earthbound cords sliding through wet soil, evolved limbless bodies independently of both reptiles and snakes. Their ancestors separated from reptilian lineages hundreds of millions of years ago, yet ecological pressures shaped them toward the same overall design. Reduced eyes, reinforced skulls, and elongated muscular bodies proved equally useful in subterranean environments, regardless of ancestry. The oceans produced another version of this repeated experiment. Sea snakes and sea kraits adapted portions of the snake body plan to aquatic life, developing flattened tails and specialized lungs for prolonged swimming. At the same time, entirely unrelated fish lineages, such as eels, evolved similarly elongated forms for moving through reefs, riverbeds, and underwater crevices. The resemblance between eels and sea snakes can appear striking from a distance, even though their evolutionary histories remain profoundly separate. This repeated emergence of limbless, elongated bodies is known as convergent evolution. Different lineages encounter similar environmental pressures and gradually arrive at comparable solutions despite sharing no recent common origin for those traits. Evolution does not move toward predetermined goals. Instead, certain body plans repeatedly prove effective under specific conditions, and natural selection refines them independently across different branches of life. Burrowing environments favor streamlined movement through confined spaces. Dense vegetation rewards flexibility and low profiles. Aquatic habitats reduce the advantage of limbs while increasing the efficiency of wave-driven propulsion. Again and again, the same pressures encouraged bodies to lengthen and limbs to shrink. Each transformation unfolded slowly across immense stretches of time. No single generation produced a limbless animal fully formed. Small variations accumulated, preserved only when they improved survival or reproduction within particular environments. Over millions of years, separate lineages drifted toward remarkably similar outcomes. The repetition itself carries scientific weight. When evolution independently produces nearly the same structural solution multiple times, researchers gain confidence that the advantages are genuine and measurable rather than accidental. Convergent evolution becomes something close to a natural experiment repeated across geological history. In distant habitats separated by oceans and eras, life kept rediscovering the same answer written in muscle, spine, and motion. Even after centuries of collecting specimens, sequencing DNA, and surveying forests across multiple continents, the story of snakes remains unfinished. New species continue emerging from the edges of scientific awareness with surprising regularity. as though entire branches of the reptile world still move quietly beyond the reach of classification. As of 2024, researchers formally describe roughly 50 to 100 new snake species each year. Some are discovered in isolated valleys or dense tropical forests, rarely visited by humans, but others emerge from places already crossed by roads, villages, and generations of biological surveys. The hidden diversity of snakes often lies not in dramatic remoteness, but in subtle physical differences overlooked for decades. Many undiscovered species are believed to inhabit tropical forest canopies, deep river systems, and remote island chains where observation remains difficult. Forest canopies alone contain layered ecosystems suspended high above the ground, environments still poorly sampled because reaching them safely requires specialized climbing equipment and prolonged expeditions. A snake, spending most of its life among branches, may remain invisible even within forests studied for years. One striking example appeared in the Bahamas during 2017. Researchers formally identified a new species of boa living on a small island already visited repeatedly by biologists. The snake had existed in plain sight, yet only detailed anatomical and genetic analysis revealed it to be distinct from neighboring populations. Discovery, in many cases, depends less on finding an entirely unknown animal than on recognizing hidden separation within familiar forms. Museums have become equally important frontiers of discovery. Shelves filled with preserved specimens collected decades or even centuries ago now undergo genetic analysis unavailable to earlier researchers. Through a process known as taxonomic revision, animals long grouped together as single species are divided into multiple distinct evolutionary lineages after DNA sequencing reveals deep genetic divergence. In effect, entirely new species emerge, not from jungles or mountains, but from drawers quietly waiting inside scientific collections. The oceans remain especially mysterious. Sea snake diversity is still poorly understood, particularly in deeper marine environments where systematic surveys remain rare. Population sizes for many species are uncertain, and large portions of their geographic ranges have never been carefully mapped. Beneath tropical waters, entire populations may persist, almost completely undocumented. Modern techniques are accelerating the pace of discovery. DNA barcoding allows researchers to identify species from tiny tissue samples, fragments of skin, or traces of environmental material left behind in water or soil. Instead of relying only on complete physical specimens, scientists can now survey ecosystems genetically, detecting hidden biodiversity through molecular signatures carried invisibly through the environment. Venom research has added another dimension to this search. Each newly identified snake species possesses its own chemical profile, shaped through millions of years of evolutionary refinement. Venoms contain combinations of proteins, enzymes, and peptides unlike those found in any other organism. Existing medicines already trace their origins to these compounds. One of the most widely prescribed blood pressure medications in the world, Captopril, emerged from research on the venom of the Brazilian lancehead pit viper through work initiated by researcher Sergio Ferreira in the 1940s. Similar studies continue today as laboratories examine newly discovered venoms for compounds capable of treating blood clots, neurological disorders, and cardiovascular disease. Somewhere in a rainforest canopy, beneath river sediment, or inside a museum jar labeled generations ago, another undiscovered species may already exist, carrying biochemical structures no human science has yet encountered. For most of their history, Snakes endured catastrophes vast enough to redraw the surface of the planet. They passed through asteroid winters, continental drift, volcanic upheaval, and repeated climate oscillations that transformed forests into deserts and shallow seas into mountains. Yet, in the span of only a few human generations, many snake populations have begun to decline at a pace far faster than their long evolutionary resilience prepared them for. A study published in the journal PNAS in 2021 examined long term population records for 14 snake species distributed across 4 continents. 11 of those 14 populations showed significant decline. The pattern appeared across different habitats and lineages, suggesting not isolated local problems, but a broad global pressure quietly affecting snakes in many parts of the world. The causes are layered together through modern landscapes. Tropical forests continue to disappear beneath expanding agriculture. Roads divide migration routes and increase mortality, and many snakes are intentionally killed by humans on site, regardless of whether they are venomous. A creature shaped over more than 100 million years of adaptation now encounters threats arriving faster than evolution can respond. Climate change carries particular consequences for ectothermic animals. Snakes do not generate stable internal body heat in the way mammals do. Their metabolism, digestion, movement, and reproduction depend on environmental temperature patterns refined over immense spans of time. As seasonal cycles shift, feeding periods drift out of alignment with prey availability, breeding windows become unstable, and hibernation timing grows increasingly unreliable. Some species face additional pressure through the wildlife trade. Rare pythons, brightly colored vipers, and island species with restricted ranges can command extremely high prices in illegal markets. A snake removed from a small, isolated population may represent not just one lost animal, but a measurable reduction in the species' remaining genetic diversity. In the southeastern United States, the eastern indigo snake has declined heavily because of habitat fragmentation and direct persecution. Once common across longleaf pine ecosystems, it is now federally listed as threatened. Elsewhere, island species stand at even sharper edges of vulnerability because their entire populations may exist within only a few square kilometers. The St. Lucia racer came perilously close to disappearing entirely. Introduced predators and habitat disruption reduced the species to a single tiny island refuge before conservation efforts intervened. The Antiguan racer followed a similarly fragile path. By the 1990s, fewer than 50 individuals were believed to remain alive in the wild. Intensive conservation work, including predator removal and habitat protection, slowly rebuilt the population to roughly 1,100 individuals by the 2020s. The importance of snakes extends far beyond their own survival. In many ecosystems, they regulate rodent populations that would otherwise expand rapidly. Rodents influence crop loss, seed dispersal patterns, and the spread of disease carrying parasites. The absence of snakes changes pressures across entire food webs, often in ways too gradual for immediate observation. An ecosystem rarely collapses all at once. More often, it loosens thread by thread. The disappearance of snakes removes a predator refined through deep evolutionary time, an animal woven into forests, deserts, wetlands, and grasslands long before human civilization emerged. Around those ancient movements, countless other living systems quietly arranged themselves. Long before engineers attempted to imitate snakes with metal joints and computer code, the animal itself had already solved a problem that machines still struggle to overcome. A snake can move through narrow tunnels, fractured stone, dense vegetation, unstable sand, flooded terrain, and tangled debris without requiring wheels, legs, or stable footing. The same body plan that once carried early snakes through ancient forests is now guiding the design of machines built for environments too dangerous or inaccessible for humans. Traditional robots perform best on predictable surfaces. Wheels require relatively even ground, and legged machines must constantly balance shifting weight across rigid contact points. In collapsed buildings or earthquake rubble, those systems begin to fail. Broken concrete, twisted steel, and unstable cavities create spaces where movement becomes uncertain. Snake locomotion approaches the problem differently. Instead of forcing the environment into stability, the body continuously adapts to irregularity through distributed motion across many joints. Engineers studying snake movement translated this principle into robotics through chains of motorized segments capable of coordinated wave patterns. These machines use the same locomotion modes seen in living snakes. Lateral undulation allows movement across uneven ground. Concertina motion enables navigation through confined tunnels, and sidewinding reduces slippage on loose surfaces such as sand or gravel. Each movement pattern emerges not from rigid steps, but from flowing adjustments distributed along the entire body. At Carnegie Mellon University's Biorobotics Lab, researchers developed robotic snakes capable of climbing trees, entering pipe systems, swimming, and traversing debris fields. Their motion algorithms were derived directly from biological studies examining how real snakes distribute muscular force along hundreds of vertebrae. The machines do not simply resemble snakes visually. Their engineering logic is fundamentally serpentine, built around flexibility rather than rigid stability. Search and rescue operations became one of the clearest applications for this design philosophy. After earthquakes or structural collapses, survivors may remain trapped inside cavities, unreachable by conventional equipment. A snake robot can move through narrow gaps between broken surfaces while carrying cameras, microphones, and environmental sensors. Its segmented body bends around obstacles instead of stopping at them. The nervous system of snakes also reshaped thinking in computer engineering. In many vertebrates, movement depends heavily on centralized control from the brain. Snakes distribute coordination across the spinal cord itself, allowing smooth locomotion without conscious regulation of every individual motion. Robotics researchers adopted similar distributed architectures in which multiple processors share local control responsibilities rather than relying on a single computational center. This increases flexibility and resilience when parts of the system encounter failure or unexpected terrain. Medicine followed a parallel path. Flexible surgical devices and advanced endoscopes increasingly borrow concepts from snake anatomy. Instead of requiring large incisions, segmented robotic instruments can travel through natural, internal pathways, curving gently through the body with far greater precision than rigid tools. In these machines, ancient locomotion becomes modern surgery. Sea snakes contributed additional lessons. Their movement through water is now studied to improve underwater vehicles used for pipeline inspection and deep-sea exploration. Even the thermal sensing organs of pit vipers inspired passive infrared technologies, later adapted into thermal imaging systems, security sensors, and guidance equipment. Each invention represents a translation across immense stretches of time. Engineers observe a body refined through more than 100 million years of evolution, then slowly convert biological solutions into mathematics, circuitry, and motion. Inside the venom gland of a snake exists a chemical archive assembled slowly across evolutionary time. Each protein, enzyme, and peptide emerge through millions of encounters between predator and prey, shaped by natural selection with extraordinary precision. What began as a biological weapon is now being examined molecule by molecule inside research laboratories, where scientists increasingly view venom not only as a danger, but as a library of possible medicines. The pharmaceutical potential of snake venom remains only partially explored. Many species contain more than 100 distinct compounds within a single venom cocktail, and most of those molecules have never been fully characterized. Some interfere with blood clotting, others disrupt nerve transmission, dissolve tissue, alter blood pressure, or interrupt cellular communication pathways. In the controlled environment of medicine, those same mechanisms can sometimes be redirected toward healing rather than harm. Several important drugs already emerged from this research. Captopril, widely prescribed for hypertension, was developed from studies of the Brazilian lancehead pit viper. Eptifibatide and tirofiban, both used to reduce complications associated with heart attacks and acute coronary syndrome, also originated from venom research. Their existence reflects a strange continuity between ancient predatory chemistry and modern cardiovascular medicine. The wider field of venom based pharmaceuticals expanded further when researchers began studying venom systems beyond snakes themselves. In 2023, scientists at the University of Copenhagen published findings connected to the Gila monster, a venomous lizard distantly related to venom bearing snake lineages. Compounds derived from its venom contributed to a new class of diabetes and obesity treatments, including the active ingredient associated with Ozempic. A reptile adapted for survival in harsh desert conditions unexpectedly reshaped modern metabolic medicine. Snake venom compounds are now being investigated for possible applications against cancer, neurological disorders such as Alzheimer's disease, and antibiotic resistant bacterial infections. Some proteins appear capable of targeting cellular pathways with remarkable specificity, acting almost like microscopic keys shaped for particular biological locks. Researchers continue searching for molecules able to interrupt tumor growth, block pain transmission, or bypass the limitations of conventional antibiotics. Obtaining venom, however, remains difficult. Milking venom from live snakes is slow, dangerous, and tightly regulated. Many medically interesting species are rare, geographically isolated, or protected. Extracting enough venom for large-scale pharmaceutical research becomes ethically and logistically challenging when relying entirely on living animals. Synthetic biology began changing that reality. Scientists can now identify the genetic sequences responsible for producing specific venom proteins, then insert those instructions into laboratory cell cultures. Instead of collecting venom directly from snakes, researchers grow individual compounds under controlled conditions. A molecule once produced inside the glands of a remote pit viper can now be replicated inside sterile bioreactors thousands of kilometers away. The paradox surrounding venom deepens with every discovery. Chemistry refined to immobilize prey and stop hearts may eventually preserve millions of human lives. One of the clearest examples remains the Brazilian lancehead itself. Before its medical significance became understood, the species was often treated as a dangerous pest suitable for eradication. Had that effort succeeded completely, one of the world's most influential blood pressure medications might never have existed. Within every venom gland still lies an immense chemical frontier, quiet and largely unmapped, carrying solutions evolution assembled long before human medicine learned how to recognize them. There is a difficulty hidden inside deep time that the human mind rarely overcomes completely. A century already feels immense when measured against a single life. 1,000 years stretches beyond memory into history. Yet the story carried inside, the body of a snake, unfolds across spans so vast that ordinary intuition begins to lose shape around them. 100 million years represents roughly 4 million human generations. Entire civilizations rise and disappear in periods that, on the evolutionary scale, resemble brief surface ripples. The transformation from small four-legged reptile to fully limbless snake unfolded across approximately 20 to 30 million years, an interval containing around 1 million generations of tiny, inherited variation. No single generation witnessed the whole transition. The change moved too slowly for direct perception, accumulating through increments almost impossibly small. An individual proto-snake never experienced itself as incomplete or transitional. It simply moved through its environment, using the body it possessed, searching for food, avoiding predators, reproducing when conditions allowed, and eventually dying. The same was true for its offspring and for the countless generations surrounding it. Evolution does not announce itself while it is happening. It operates quietly through differential survival repeated across unimaginable spans of time. Leg loss was not guided by foresight. No organism intended to become a snake. Bodies that happened to move more effectively through confined spaces left slightly more descendants. Subtle elongation of the torso, marginal reduction of the limbs, small changes in skull flexibility or sensory processing, each adjustment carried consequences measured not in dramatic leaps, but in probabilities. Over millions of repetitions, those probabilities accumulated into anatomical transformation. The same patient filtering shaped nearly every complex structure in vertebrate history. Eyes emerged through gradual refinements in light-sensitive tissue. Wings developed from limbs that originally served entirely different purposes. Hands capable of building instruments began as ordinary grasping structures carried by climbing mammals. The brain itself reflects layered modifications built upon far older nervous systems inherited from distant ancestors. Understanding this process does not reduce the wonder of what emerged from it. The opposite happens. The snake becomes more extraordinary when viewed not as a simplified reptile, but as a body refined continuously across 100 million years of environmental testing. Its structure endured because it worked with remarkable consistency across forests, deserts, rivers, grasslands, caves, coastlines, and oceans. Every living snake still carries traces of that ancient transformation hidden within its biology. Some species retain remnant pelvic bones suspended deep inside the body. Embryos briefly begin constructing legs before the developmental program shuts down. Silenced genetic pathways remain preserved in the genome like dormant architectural plans from another era. The history of lost limbs was never erased completely. It survives quietly beneath the scales. And despite abandoning the appendages most vertebrates rely upon to climb, dig, grasp, or run, snakes spread across nearly every continent except Antarctica. They entered trees, oceans, underground burrows, mountain forests, floodplains, and coral reefs. A lineage that surrendered arms and legs eventually occupied almost every ecological space available to a cold-blooded predator. The body that seems reduced at first glance became one of evolution's most enduring designs, shaped not through urgency, but through the immense patience of time itself. There are animals whose bodies feel immediately understandable to us. Mammals carry faces that resemble our own patterns of expression, limbs that move in recognizable ways, eyes directed toward familiar goals. Snakes stand outside nearly all of those intuitions. Their bodies seem to remove features humans unconsciously associate with intelligence, communication, and agency. No hands grasp the world around them. No voice shapes sound into warning or attachment. No visible expression moves across the face. And yet snakes persist across deserts, forests, oceans, mountains, wetlands, and underground systems with astonishing success. Their existence suggests another possibility for life, one that does not depend upon the traits humans instinctively prioritize. A snake experiences its environment through sensory channels radically unlike our own. The flickering tongue, gathering particles from air and soil, is not merely detecting odor in the simple sense humans understand it. Each movement samples chemical traces layered across space and time. A snake moving through a forest is reading invisible currents left behind by prey, predators, water, decay, reproduction, and movement. The world arrives not primarily as image, but as chemistry. Ground vibration adds another dimension. Without external ears, snakes detect subtle mechanical disturbances traveling through soil, wood, sand, or stone. A passing animal creates shifting patterns long before it becomes visible. To a snake resting against the earth, movement itself becomes part of the landscape. The boundary between touch and hearing softens into something difficult to translate into human terms. This difference is not deprivation, it is specialization. Human perception privileges distance, vision, and symbolic communication because those traits served the evolutionary path of primates. Snakes followed another route entirely, refining systems that allow survival through concealment, stillness, precision, and metabolic efficiency rather than endurance or social coordination. For a long time, popular culture reduced snakes to instinct driven automatons, creatures imagined as little more than reflex and appetite. Research increasingly complicates that picture. Biologists studying snake cognition observed forms of learning, spatial memory, and behavioral flexibility inconsistent with the stereotype of a purely mechanical predator. Some species can remember environmental layouts, adjust hunting strategies based on previous encounters, and recognize recurring stimuli over extended periods. King cobras offer particularly intriguing examples. Unlike most snakes, they construct nests by gathering vegetation into mounds where eggs remain protected during incubation. Females may remain nearby, guarding the nest for significant periods. This behavior requires environmental assessment, spatial organization, and prolonged investment beyond immediate feeding or reproduction. Scientists are still attempting to understand the full behavioral complexity involved. Yet there remains a boundary. Science cannot cross completely. Fossils preserve bone, genetics preserves inheritance. Neither preserves subjective experience. Consciousness leaves no direct physical imprint recoverable through excavation or sequencing. The interior life of a snake remains inaccessible in any complete sense. Still, the question lingers quietly beneath the biology. What does existence feel like inside a body so different from our own, guided by chemical gradients instead of language, by vibration instead of conversation, by heat traces drifting through darkness rather than reflected light alone? A snake moves through the same physical world humans inhabit, encountering hunger, danger, reproduction, shelter, and death. But it meets those realities through an entirely separate architecture of sensation and behavior, one evolution assembled without ever consulting the assumptions of the human mind. Across the history of life, change is often imagined as accumulation. More limbs, more speed, more intelligence, more complexity. Human language naturally drifts toward the idea that evolution builds by adding. Yet the body of a snake tells a quieter story, one written not through expansion alone, but through subtraction refined over immense stretches of time. The snake became what it is through disappearance. Legs vanished first in fragments, then entirely. The ancient skeletal anchors that once supported walking dissolved generation by generation until only hidden remnants survived in a few lineages. External ears faded from the body. Eyelids fused into transparent scales. The compact torso, typical of many vertebrates, stretched outward into a chain of vertebrae and muscle unlike any other major terrestrial predator. At nearly every stage, structures familiar to other animals receded instead of multiplying, and still the lineage flourished. The disappearance of legs opened physical spaces inaccessible to many competitors. Burrows, root systems, narrow crevices, dense undergrowth, and underwater passages became navigable through elongation alone. A body, no longer interrupted by limbs, could move with extraordinary continuity across surfaces where feet became obstacles. Other losses reshaped perception itself. Without dependence on external ears, vibration traveling through earth and water gained greater significance. Chemical sensing expanded into a primary map of the surrounding world. The forked tongue and Jacobson's organ formed a partnership capable of tracking invisible molecular trails with astonishing precision. The sealed spectacle, covering the eye-protected vision while allowing uninterrupted contact with dust, debris, and soil. In evolutionary biology, reduction is often misunderstood because human intuition associates loss with damage or decline. Yet natural selection does not preserve structures because they appear impressive. It preserves what continues to function effectively within a given environment. When a feature becomes costly, unnecessary, or limiting, evolution may diminish it over millions of years, freeing energy and anatomy for other adaptations. This pattern appears throughout the vertebrate story. Early mammal ancestors gradually reduced large jaw bones that reptiles once used for chewing. Those bones eventually became the tiny auditory ossicles of the mammalian middle ear, delicate structures capable of transmitting subtle vibrations that later allowed mammals to perceive intricate soundscapes. Wings evolved in part through modified forelimbs. Whale hind legs receded as bodies adapted completely to marine life. Evolution repeatedly transforms by reshaping and simplifying as much as by enlarging. The snake represents one of the clearest examples of this principle carried to an extreme. It did not become remarkable by collecting every possible trait. It became remarkable through ruthless specialization. What remained after each reduction was refined with extraordinary precision until the resulting animal could inhabit forests, rivers, deserts, oceans, mountains, grasslands, and underground tunnels across nearly every continent on Earth. There is no moral hidden inside this process, no instruction waiting beneath the biology. The transformation occurred without intention, guided only by differential survival across uncountable generations. Yet the pattern remains quietly striking. A lineage that surrendered limbs, external ears, movable eyelids, and much of the familiar vertebrate body plan became one of the most widespread and enduring groups of predators the planet has ever produced. Long before human language formed its first written symbols, before cities rose beside rivers or stone walls crossed deserts, the snake lineage was already ancient. Its history reaches back at least 128 million years into a world where flowering plants were only beginning to spread and enormous reptilian bodies still dominated much of the land. Since then, the surface of Earth has rearranged itself repeatedly. Continents that were once joined drifted apart at the pace fingernails grow. Mountain ranges lifted from shallow seas and slowly wore back down beneath rain and wind. Through every rearrangement, snakes remained present, moving quietly through landscapes that no longer exist. The scale of that duration resists ordinary comprehension. Human civilization measures itself in centuries. Recorded history extends roughly 5,000 years into the past, a span that feels immense when viewed against individual lifetimes. Yet, compared to the age of snakes, those millennia nearly vanish. The pyramids at Giza, weathered by more than 4,500 years of desert air, occupy only the thinnest fraction of the snake timeline. Entire empires rise and disappear in intervals smaller than the lifespan of many geological formations. And still the lineage continues. The snakes alive today are not unchanged relics from the Cretaceous world. Every surviving species has been shaped continuously by mutation, selection, migration, and extinction across incomprehensible numbers of generations. Ancient forms disappeared while new ones emerged. Venom systems diversified, senses sharpened or diminished depending on habitat. Bodies adapted to forests, coastlines, rivers, grasslands, caves, and oceans. Yet, beneath all that variation, the central architecture endured with remarkable stability. The elongated spine, the absence of limbs, the kinetic skull, the chemical reading of the environment through tongue and organ, all persisted through climates and catastrophes that erased countless other lineages entirely. This persistence is not evolutionary stillness. It is evidence of a configuration so flexible that it remained effective across radically different worlds. The same fundamental body plan moved through humid greenhouse forests of the Paleocene, through colder epochs shaped by expanding ice, through tropical wetlands and drying continental interiors. Few vertebrate designs have endured so long while requiring so little structural revision. There is something quietly disorienting about measuring human existence against such continuity. The modern world feels immediate and permanent because it surrounds us completely. Roads, buildings, electrical grids, satellites, and digital systems dominate perception. Yet, from the perspective of deep evolutionary time, these belong to a moment so narrow it is almost impossible to represent proportionally. A snake crossing a forest floor today carries within its anatomy solutions refined across spans of time, vastly older than agriculture, older than language, older than our species itself. The animal does not know this history. It moves through heat, scent, vibration, hunger, and shelter exactly as its nervous system allows. But the history remains present all the same, written invisibly into bone structure, embryonic development, sensory pathways, and genes inherited through millions upon millions of uninterrupted lives. We experience time in decades. Evolution experiences it in continents, oceans, and extinctions. Against that scale, the present becomes very small, a brief surface ripple moving across a lineage that has already outlasted worlds. A snake crossing through grass appears simple at first glance, almost minimal. A narrow body, a flickering tongue, movement so quiet it seems detached from effort itself. Yet beneath that surface exists a biological history older than mountains, still rising today. The animal moving through sunlight carries inside it more than 100 million years of uninterrupted survival, a continuity extending backward through vanished forests, drowned coastlines, and worlds populated by creatures long reduced to stone. Inside the skeleton of many snake species, traces of another anatomy still remain. Deep within pythons and boas, Tiny pelvic remnants float disconnected from the spine, silent echoes of limbs that once pressed against ancient ground. These structures no longer support walking, yet they persist across immense spans of evolutionary time, repurposed rather than entirely erased. The body remembers even when function changes. The memory extends deeper still, beyond bone and into the genome itself. The genes required to construct limbs were never completely removed. They remain embedded in sequences of DNA inherited through millions of generations. What changed were the regulatory systems controlling them. Molecular switches altered gradually through selection until the developmental instructions fell quiet. The blueprint survived even after the architecture disappeared. That hidden history briefly resurfaces each time a snake begins life. Within developing embryos, small limb buds emerge for a short interval before fading away. Under magnification, the process becomes visible with startling clarity. The body initiates an ancient developmental pathway, then interrupts it almost immediately. In a matter of days, structures that once would have become legs recede back into the growing form. Evolutionary history flickers through embryonic tissue and vanishes again before birth. The connection between snakes and humans reaches beyond anatomy and into the nervous system itself. Long before cities, agriculture, or language, primate ancestors shared landscapes with venomous snakes. Across evolutionary time, individuals capable of rapidly identifying serpentine shapes possessed a survival advantage measurable in offspring and generations. Modern neuroscience now shows that the human visual system responds to snake-like forms with unusual speed, activating specialized pathways through regions such as the pulvinar nucleus of the thalamus. The reaction occurs before conscious thought fully assembles. In this way, the snake exists partly inside the architecture of the human mind. It also exists within medicine, chemistry, mythology, and culture. Venoms evolved to immobilize prey, now contribute to treatments for hypertension, clotting disorders, and chronic disease. Symbols of snakes appear independently across civilizations separated by oceans and centuries, emerging repeatedly in stories, rituals, and art. The lineage wound itself through human history long before humans understood what it was seeing. And still the animal remains fundamentally itself. No limbs returned, no external ears reappeared. The body stayed narrow, elongated, and close to the earth. What might appear at first as reduction became refinement carried to extraordinary precision. Chemical sensing sharpened, the jaw expanded its mechanics. Movement diversified into forms unmatched by limbed vertebrates. Nothing about this transformation happened quickly. It unfolded through countless generations living ordinary biological lives, each one adding an almost immeasurable adjustment to the next. Yet across enough time, tiny changes accumulated into one of the most distinctive body plans ever produced by evolution. The legs disappeared, the lineage endured, and what endured proved sufficient to cross continents, survive extinctions, and remain present in the world even now, moving quietly through grass while ancient history travels within every scale.